Hi, my name is Dana Mann. I'm a Jacksonville State University graduate student here in Jacksonville, Alabama. And today my lecture topic is squid bacteria bioluminescent symbiotic relationship with its bioluminescent bacteria Vibrio fisheri. Now, quick overview of bioluminescence is that bioluminescence, if you don't know, is the illumination of light from a organic source. It could be from bacteria, phytoplankton, squids, jellyfish use it. For instance, you have an anglerfish that is able to produce it from its antenna off its head. Also, some insects do it. Most notable example is a glowworm, or fireflies. But its primary function is usually communication by attracting males, like in the glowworm. Also, it can be used to find prey. Like the anglerfish, uses it to attract its other fishes to it in the deep sea. But it could also be used for defense, which is what my discussion is, as I'll get to in a second. Now, there's two types of bioluminescence. I don't want you to get this confused with some like the glowworm that use, or a anglerfish that use a photosphere, which is a luminescent spot produced by their light organs. That's different from a symbiont with Vibrio fisheri and the squid, in that it's a bacterial symbiont. And the bacterial symbiont is housed in its host squid that provides a light source. Now, first, how was this relationship initially started? It's horizontally transferred from parent to offspring with bacterial selectivity. The way that Vibrio fisheri is able to invade, not so much invade, but house and take up residence inside its squid host, in that it's gram-negative, which is very selective in the squid. It's able, a gram-negative is able to penetrate into it, as I'll show you in a second. There's also a couple other things. Inhibitory meniscus, which helps collect the bacteria better. There's also the antenna on the squid, or the sort of muscles, the arms of it that help collect it in better. But it also helps house it in the housing. You'll see the acids a little bit better and keeping other bacteria away, but it allows only Vibrio fisheri to house it. But even closely related bacteria to Vibrio fisheri sometimes are able to infect the squid, but it's never there for long. Usually the native Vibrio fisheri will outcompete it eventually. Now, the symbiotic relationship and function has basically a light organ within the mantle of the squid. It helps direct light. There's filters. It's in a similar context like the eye, in that it's able to focus. There's filters. It works in that way. But its function is that it's able to produce a silhouette at the bottom of the squid, making it nearly invisible to predators below. As you'll see in the next slide, pretty much it will show a silhouette. In this case, this isn't exactly the squid, but as you see in this example, a prey could easily see that, even with its very primitive eye, unlike ours, which is very complex. But when you come over here, even though we can see it, a prey with its, like I said, its primitive eye, very simple, cannot tell between the organism and the background of moonlight or sunlight behind it. For the host, its common name is Hawaiian bobtail squid, It's nocturnal, as I'll show you in a second. Is that it expels its bacteria symbiont, the fisheri, in the very early morning. Then while it's asleep, it builds it back up, and it has two other defensive mechanisms. It's able to release ink in a distraction or as a decoy, as well as able to bury itself in the sand. Now, it keeps the bacteria symbiont throughout its life. In fact, it's probably not able to live without the symbiont. It's also a very interesting relationship because unlike other symbionts with bacteria like Wolbachia, Buchnera, a lot of times this one is able to live outside its host for extended periods of time. In fact, fisheri is seen freely in the environment, much more in areas where of course the bobtail squid live. But it's very minute, but it's in free swimming in marine environments. But what's very interesting is that it's able to be manipulated for in research. Both of them could be manipulated separately. All right. Now, in this slide, as you can see, pretty much the bacteria population throughout the a 24 60-hour period is first over here you have, of course, the bobtail squid goes to sleep, in which case the bobtail fisheri bacteria raises in population until nighttime, where it hits a peak. And at that peak, of course, as the squid hunts, it collects its food, and then it expels again about 90% of the bacteria. And from there, when it's asleep, it'll raise its bacteria symbionts again till it gets to a peak quorum sensing population, and then again it'll go hunting at night until the wee morning before it goes to sleep. It'll expel it all out in one final flush, and then it repeats the cycle over and over again until it dies. Now, quick fact sheet about Vibrio fisheri is that it's a marine gram-negative rod-shaped bacterium, free living, of course, in the marine, but very, very small, and it is quorum sense to bioluminesce. What that means is, even if you had some here and some there, they won't produce any light. But once you put them together, that's when they'll start emitting light. Once they hit a optimal population in a certain concentration. Right? Now, its relationship between the bacteria fisheri and the bobtail squid host is very unique. The way it selects its host, fisheri selects its host, or I guess you could say how the squid selects its bacteria symbiont, is that first it has very strong flagella that gather all bacteria into its light cell. This is during the initial stages when the bobtail squid is very young. It's able to use its flagellate appendages to get as much bacteria as it can, just to get it close to its light organ. Then it will secrete a mucus. Once the flagella sense the right bacteria, the right gram-negative bacteria, it'll start secreting mucus, which allows the gram-negative to collect closer together, clump up together. And as it gathers into the light organ, the fisheri are able to be resistant to the hypothalamus, the substrate that is pretty much takes away oxygen. Because in that light organ, it is very toxic to most bacteria, except for Vibrio fisheri, which is able to gather it up and use it, produces it. Yeah. Now, how is it able to, I guess, come into those light sensing organs is not completely understood. uh, with in terms of light production. But I think in future, of course, research they will find an answer. Now, like I said, here is the developmental change in the adult light organ. Of course, during the very initial stages, within the first couple days of the bobtail squid, it has these appendages close to its mantle right here. That over time, in the first couple of days of its life, it will attract the fisheri, and then eventually they will break off, like right here. And in these two slides, you see is that you'll see, yes, the staining of the fisheri as it comes in contact with the light organ. But as you can see, there's apoptosis, lysis of all of the appendages, the flagellate, start to die, so it breaks off. So that, of course, it's not unnecessary. Now on this slide you have the initial interaction of juvenile host with the environment. Like I said, yeah, after it's collected by the flagellate and they gather it up, first you'll see it sort of trends in selectivity. First you have it gathers practically any bacteria, simion, and then it starts to get more and more selective as it goes down the line and over time, until finally, ultimately, only Vibrio fisheri houses in the light organ and is able to produce the light for its squid host. Now, I hope that you enjoyed this talk. Again, check out my other discussion with Wolbachia and Bacterial on this channel. Be kind to give me a thumbs up and leave a comment below. Alright, thank you for watching.